The microphone is okay, right? All right. Well, thanks, Holden, for inviting me to talk. I'm going to share with you today a technology that's hopefully going to get published pretty soon. And what we've enabled is the pairing of TCR alpha-beta sequencing with multi-parametric phenotyping in single T cells. So hope to tell you a little bit about it, what it can do, and hope to convince you how great it is. So in terms of single-cell analysis, I really don't think I need to explain to a room full of people at Stanford the benefits of single-cell analysis as opposed to bulk analysis, where you analyze a group of diverse population of individuals individually rather than taking the whole and basically taking an average. And this is Time Magazine's rendition of the average American that they published a few years ago. And it's been well established now that single-cell analysis can reveal important information that's masked in bulk analysis. And T cells in particular are very, very complex. As we know, there are many types of T cells. There's CD8 T cells and CD4 T cells. And really, this is just scratching the surface. As T cells, we know, can differentiate into many different diverse functions and phenotypes. And in analyzing a population of immune cells as a whole, you're really not doing this individual complexity any justice. So, Stanford has pioneered multi-parametric analysis, as Holden mentioned, in terms of the Fluidigm technology and also the CyTOF technology, which enables, rather than looking at 4 or 5 or 10 different parameters on single cells, which we can do by fax, we can look at 40 or 50 different parameters, or in the case of Fluidigm, maybe 96, theoretically, different parameters on single cells, and we can really get to appreciate the diversity apparent in T cell function. But one serious limitation to these technologies, which I've used, is that they seem to ignore the most important molecule or gene that the T cell expresses, and that's, of course, the T cell receptor. And we know that the T cell receptor is basically everything in terms of how the T cell functions. It mediates its selection, it mediates immunity, it mediates tolerance. So we really are missing out on the T cell receptor through these technologies, which can basically tell you whether it exists or not, but really can't tell you much else about it. And another thing about the T cell receptor is its theoretical diversity, which is, I don't know, 10 to the 12th or 10 to, I don't know what the latest estimate is. But really, if 2 cells have the identical T cell receptor, you can pretty much conclude that they came from a common precursor somewhere along during development. So if you look at an immune response and you're also able to look at the T cell receptor sequence, it gives you a lot of additional information. Not only does it give you a snapshot of the immune response at a given point in time, but given the T cell receptor sequence, it gives you a really strong idea of how that immune response evolved, because you can look at the clonality. Additionally, by knowing what the T cell receptor is, it enables you to do a lot of functional studies afterwards, for instance, find ligands and whatnot. So this is basically an outline of the technology I've developed. I apologize, I didn't develop some fancy machine that vaporizes cells and does mass spec or pipettes, obscenely small volumes with high accuracy. It's really quite simple, and I think that anybody in this room with marginal training could learn how to do this. And what it involves is, so the first aspect we developed is a TCR sequencing part. And it involves a series of nested PCR reactions, which ultimately enable analysis on the Illumina platform. And we developed this technology a couple years ago and published a few papers with it. And we got it working quite efficiently. So more recently, we thought, well, what about phenotyping cells with the same kind of strategy? So rather than just take the TCRs and sequence them and tell you what the TCR alpha-beta sequence is, why not throw in some cytokine and transcription factor primers and do the exact same thing? So we do reactions with cytokine and transcription factors, nest them, which we found was absolutely crucial to get into TCRs to work, do a nested PCR, and then analyzing it in the end. So basically, the readout is you take a barcoded well through Illumina sequencing, and it'll tell you which TCR sequences are there and also which cytokines that particular cell is expressing. And so the third reaction after the first 2 nested reactions is actually where we incorporate the barcodes. So the third reaction we do in 96-well plates, and we have 12 different barcodes here for the columns and 8 different barcodes that specify plate and row. And you have to use a different set of 8 barcodes for each subsequent plate. But what ends up happening is when you pipette them this way, of course, every cell ends up with an individual barcode that specifies the plate, the row, and the column. So based on this, you can put about 25, 30 different plates on one reaction pretty easily and get data from 2,000 to 3,000 cells using one Illumina run with current technology. And this current technology is really expanding. So once we get the Illumina reads, basically now you have 25 million reads or so, and you have barcodes on each particular cell, and you have cytokine transcription factor sequences as well as TCR sequences. Then we go through the bioinformatics pipeline, where we take the raw reads, we go through a bunch of filters by quality. We find a consensus because right now we're using paired end technology, so we're sequencing from the front and reverse end, so we find a consensus in the middle to match up the reads. And then we have the barcodes, front, column, row, and plate, of course, and then we map them to each individual well, and then in each well we resolve the TCR sequences and cytokine expression. So we got this working quite well, and we're actually very highly efficient. So the paired TCR sequencing works at about 90% efficiency. We get 90% efficiency on TCR alpha and beta, which I think as far as we know, the highest reported efficiency for single-cell TCR sequencing. Another important aspect of this that I actually should have mentioned, but I don't, is that, I don't have a slide for it, but that we can pull out multiple alpha chains from each individual T cell. And this is actually very important because it's been long established that T cells don't undergo allelic exclusion at the alpha locus. So individual T cells can, in fact, express 2 alpha chains. And oftentimes, one of the alpha chains is non-productive, but you can even express 2 productive alpha chains. So really, people who do sequencing without using single molecule sequencing technology can frequently be misled as to what the TCR is. And before we started doing this kind of sequencing, frequently by Sanger sequencing, I would see that one T cell clone always expressed one non-productive alpha chain. I would never get the productive alpha chain from it. So this really is a significant advance, and I think that in terms of TCR sequencing in single cells, you really, really need to do single molecule sequencing in the long run to get a truly accurate TCR alpha and beta. So in terms of the phenotypic analysis, so how accurate are we? People who use biomark on single cells in the past probably know that one limitation of biomark is that although it can quantify things, it doesn't always amplify what you're looking to amplify. So when I've done it on TNF or interferon gamma, I would get terrible efficiencies where I would amplify about 10% of the cells. Now, biomark could tell you what the quantity of that TNF and interferon gamma signal might be in those cells. It can quantify it in relation to actin, perhaps, but really you're missing most of the cells that actually express the gene. So we tested the accuracy of our phenotyping technology by using these cytokine capture assays by Miltenyi. So we couldn't use intracellular staining because then we couldn't do our reactions because of the cell fixation. But Miltenyi sells these kits where basically capture an antibody on the surface without fixing the cell. So they have all sorts of, they have interferon gamma, TNF, and various interleukins available. So what we did was we did this kind of analysis where we used the Miltenyi kit, stained it with interferon gamma, for instance, and we basically sorted positive and negative populations for each individual cytokine, and then put it through our assay. So sorted the single cells and looked at what kind of readout we get through our analysis. And you can see that we get really highly accurate reads in comparison to the Miltenyi kits. So for instance, this is interferon gamma, where we take the positive population and basically we can hit interferon gamma signal in 92% of the cells. And in the negative cells, we get no signal whatsoever. So we validated this for 6 different cytokines, and we get 80 to 90% on average hitting the positive here. And we get very, very low false positive rate, which I'm not going to show you the data, but we think that these cells are really cells that do express the genes, but for some reason are not expressing the protein. So we get really high accuracy on all the cytokines we've tested. And of course, not only, for instance, do you get interferon gamma, but we can get 17 other parameters as well, including transcription factors and all sorts of other cytokines. And in terms of transcription factors. Now, people who have done staining for transcription factors know what a pain it is. So you actually need extra fixation steps to adequately quantify transcription factors. And I know from our experience, for instance, with FOXP3, it's a pain to get to work by CyTOF. And it's really not even that easy by flow cytometry. And other transcription factors like TBET, GATA3, RORC, forget it. Forget trying to get all of them in one particular cell. And I think that we can do these pretty well. So this is FOXP3 analysis. So it's tough to validate transcription factors because you don't really get cell surface markers that can recapitulate transcription factor expression that easily, with one exception being FOXP3. So we sorted CD25 high populations and CD25 low populations. And you could see in the CD25 population, we're getting FOXP3 90% of the time. In the CD25 low population, we never get FOXP3. And of course, we get all these other transcription factor signatures in these single cells at the same time. And when we compare the FOXP3, for instance, the flow cytometry, we're basically every bit as accurate as flow cytometry. In fact, there's a greater overlap you can see between the CD25 high and low populations and flow, whereas in our method, they're really nicely separated. So to summarize then what we can do with this technology, the advantages and disadvantages. So I think that we're the only technology that can do paired TCR sequencing plus phenotyping in a high throughput manner at the same time. I guess you can do it with biomark, but it gets really costly. We can do about 20 plates with relative cost effectiveness. I think I calculated it at right now with the current technology, maybe $1 or $2 per cell. So we can do this. I don't think any other technology can offer this right now. In terms of TCR sequencing, important aspect of doing single cell as opposed to bulk is that the PCR bias doesn't affect results. So it's easy. There's a lot of people have used bulk deep sequencing technology to look at TCR repertoire, but in those cases, you always have to be careful that you're not amplifying one particular clone better than another, leading you to draw false conclusions about clonality. And really, in terms of single-cell analysis, obviously, you don't need to worry about this. It enables many parameters that cannot be done through CyTOF or FACS. So the example I bring up is transcription factors. You really can't, even by FACS, you really can't look at 5 or 6 transcription factors at the same time. And I think we can do this pretty accurately. Another thing we can do is that we can actually look at splice variation and polymorphism because we're actually getting the sequence. So unlike technologies like biomark, where you're basically looking for some amplicon, we actually can look at the sequence. And I've seen in our analysis, for instance, we can detect, for instance, IL-2, we can detect 3 different splice variants of IL-2. Now, I don't know what the physiologic relevance necessarily is, but if someone were interested in looking at that, they could. We could also detect single nucleotide polymorphisms. I've definitely seen situations where we've had single nucleotide variation in different transcription factors. And we think that it's very efficient, and of course, it's pretty cost effective in relation to other technologies. Now, there are some limitations, of course, that I'll acknowledge. One, it's not quantitative. So, although we can get a read number for every individual parameter per cell, we do not intend that to be meant to quantify relative read number because our technology is basically designed to saturate the signal. So if you see a transcription factor there or a cytokine there, it's pretty much all or nothing. It's either there or it's not. And of course, we do not detect protein expression. So people can argue that you're looking at RNA rather than protein, and we know that they can clearly be discordant. A lot of genes are regulated translationally, so that's a serious limitation as well. But given that, I think that in terms of overall, I think that it really does enable us to do things that we really couldn't do before. So now what about biology? And so I'm going to tell you a little bit about how we apply this and a little bit about what we can do with this kind of technology in terms of looking at biology. So what we started to look at was, we said, okay, let's pick something to look at and see what we can learn about this using our technology. And we chose analysis of tumor-infiltrating T cells. And we know they're important. As a lot of you guys probably know, T cells, T cell therapy for tumors was Science's breakthrough of the year last year. And we've now established that T cells are very, very important in mediating tumor immunity. And there are some exciting drugs out there, PD-1 and CTLA-4 antibodies that are specifically designed to incite anti-tumor T cell responses. And so in terms of tumor immunity, I started to look at colorectal cancer. And colorectal cancer, thanks to the work of many, including Bert Vogelstein, is actually the textbook example of how a tumor progresses from a benign growth to a malignancy, presumably through a series of mutations that it accumulates along the way that hinder its ability to regulate its own growth. So in terms of the immune system, I think that the traditional thinking was that somehow the immune system was there to check this progression and clear away cells that have accumulated mutation. But I think that a more recent concept that's really quite becoming better understood now is that in some cases inflammation might be actually promoting this progression. And we know that in certain mouse models of tumors, IL-17 secreting T cells, actually, if you knock out IL-17, you quicken this progression to tumorigenesis. We know from human tumors that in many cases IL-17 is a poor prognostic indicator. So people that have higher degrees of IL-17 secreting T cells have poor prognosis in their cancer. And as I'm also a gastroenterologist, I didn't tell you that about myself, but there's a classic example in medicine where inflammatory bowel disease, which is a disease characterized by inflammation of the bowel, you have a well-known increased risk of colon cancer. And this risk of colon cancer is directly proportional to the extent and your duration of your disease. So this really strongly indicates that inflammation might be doing something, not only to keep tumors from forming, but actually promoting tumorigenesis. So what we decided to do was we started to look at tumor-infiltrating lymphocytes that we obtained from colon cancer specimens through Stanford Hospital. And we started to look at phenotyping through 17 different parameters, as well as TCR sequencing analysis. So here's what we learned. So this is a PCA, basically distilling diversity of these different types into 2 different parameters. So don't worry too much about particulars of PCA if you guys aren't too familiar with it. All you need to really know is cells that are next to each other pretty much are similar in phenotype, and cells that are far apart from each other are different in phenotype. And we can clearly see that when we compare, so we compared 3 different groups of T cells. We compared T cells derived from the colorectal cancer itself, T cells derived from normal colon, which was resected in the same patient, so the patient gets his colon out, and we take T cells from the adjacent normal colon as well as the tumor, and T cells from the peripheral blood. And you can clearly see that these T cells assume very different phenotypes. So there is a lot of overlap, but you can see that the T cells from the tumor all generally happen to be grouped over here, whereas T cells from the normal colon tend to be grouped down here, with the peripheral blood clearly assuming a different phenotype. So just basically illustrates in one snapshot how different these 3 populations of T cells are. So what more can we learn? So in terms of, we get a lot of information, obviously. So this is basically this tumor-infiltrating lymphocytes. And a few things that we can see is clearly that there's a lot of clonal expansion going on. Okay, so each color bar represents 1 TCR. Whereas in the stimulated the colon sample adjacent to the tumor, really strikingly, there's only 4 T cells in the whole set that are expanded, and they're only expanded to they're only expanded once. In other words, there's 2. You find that TCR twice in the sample rather than once. But here you get massive amounts of clonal expansion with certain T cells accounting for about 10% of the whole population. And you also see, not only can you see the amount of clonal expansion, of course, but you can also look at a phenotypic profile on each particular T cell clone. So here we show how we can link TCR specificity to phenotype. Now, another thing we understand is that we can see from our analysis that even tumor-infiltrating lymphocytes, although I showed you they cluster in a similar location on the PCA, you can see that they're quite heterogeneous. So you see that, for instance, this clone is different than this clone, which is different than this clone, and I can show you a little more about this later. For instance, this expanded clone largely does not express interferon gamma, whereas this does. And you can also see that if you look at unexpanded versus expanded and you cluster them differently by phenotype, you can see that certain phenotypic groups are more present in unexpanded versus expanded clones. So I showed you a lot of fancy figures, so what about biology then? So here's where I'll go into a little bit about specifics of what we can learn. So we look more carefully at the role of FOXP3 and RORC, which is the gene that encodes ROR gamma T. We look more carefully at this in colon cancer for a few reasons. One, the function of FOXP3 is controversial. So people have reported that FOXP3 can be a positive or a negative prognostic indicator in tumors. So this has been a source of confusion for a long time. However, as I mentioned earlier, the role of RORC or ROR gamma T, I use them interchangeably, and IL-17 is actually associated with tumorigenesis and poor outcomes. So you have the FOXP3, which people really don't know exactly what they mean, while we have the IL-17 producing cells we know promotes tumorigenesis. So looking more carefully at these markers then, we know that certain markers in these cells are biased in expanded versus unexpanded clones. So when you look at these expanded TCRs here, you see a higher percentage of them express ROR gamma T and IL-17 as opposed to the unexpanded T cells here. And we also know that the higher proportion of these seem to express FOXP3 than the expanded clones here. And we know this is statistically significant. And one striking thing we looked at, we learned when we looked at FOXP3 positive cells, is that they cluster into two distinct phenotypes. So it's a heterogeneous population if you look at FOXP3 positive cells. And one population co-expresses ROR gamma T, and one population does not. And a few striking differences about these two populations. Not surprisingly, the RORC positive cells, basically most of them, just about all of them express IL-17, whereas the RORC negative cells, you see almost none of them express IL-17. Another really striking aspect of this is that the RORC positive cells were basically all clonally expanded, whereas these cells, you only see their TCRs once in the whole data set. So now you start to see what we can do by looking at the TCR sequence in addition to the phenotypic analysis. So basically, it lets us know this. It lets us know that the FOXP3 positive cells that don't express RORC generally do not express IL-17, are generally unexpanded, whereas these double positive cells are usually expanded and they do express IL-17. So then we look more carefully. So we know that these cells are clonally expanded. So then we looked, okay, so what about the TCRs that share their TCR sequences within the rest of the data set? And what do those cells look like? So basically, these are the population of cells that have the same sequences as these FOXP3 RORC double positive cells. And we looked at their phenotypes. And of course, they don't express FOXP3 because otherwise they'd be here. But these cells and these cells share sequences. And we know that, and we can see that they basically look very similar. They express RORC and IL-17. They have a lot of TNF and interferon gamma as well. So these are the typical, what we call Th17 cells, because they assume this phenotype where they express ROR gamma T and express IL-17. And we know that they're very highly clonally expanded within this subset, and we know that they share TCRs with this group of cells. So now what we can see is that these cells share a common ancestry, which would not be possible without looking at their TCR sequences. Another thing is we can look more deeply into their TCR sequences. So by looking at analysis, so here's an example of IL-17 producing cells that basically have the same TCR. So another tool we can do is that we can look at different TCR sequences and see how similar they are to one another, implying convergence and seeing if they possibly recognize discrete antigens. And here's a striking example where these two TCRs in a set of like 400 T cells were highly expanded, one making up 52 out of those 400 cells and the other making up 8. So these are two of the most highly expanded TCRs. And we see that these two TCRs basically have the same exact TCR alpha chain, which, and we know that they're different here because they utilize different nucleotide code, but somehow these have converged upon the same exact alpha chain sequence. And this really is not very likely by chance, especially in this TCR where you have 12 nucleotides that are randomly added by N nucleotide addition. So these 4 different amino acids were not germline encoded. So this basically occurred by chance in both these TCRs. And if we look at the beta chains, basically similar thing happened. They're identical with the exception of 2 amino acids, and they arose through very, very different BDJ recombination events. So this is really strong evidence that these 2 highly expanded TCRs that make IL-17 within this tumor basically arose through stimulation by the same MHC peptide. So in terms of what do we learn from this? So we can start putting together this data into a story. So we learned, this pointer is very confusing. I keep turning it in my hand. Okay, so we learned that you have these Th17 cells, which are highly expanded within the tumors. And we know that these FOXP3 positive IL-17 cells are also expanded. And actually, people have seen both these groups of cells within the tumor, but really nobody could know whether they arose from the same T cells. And now we've shown, basically, that one population has clearly led to another because we know that they share the same TCR sequence. Another thing we've shown is that they seem to be stimulated in an antigen-specific manner, so they're not just bystanders that are passing through that just happen to hit inflammation and expand. We know that because we can show examples of TCR sequences within these within this subset of T cells that actually show strong evidence of convergence, meaning they were selected by the same antigen. So this was also something that was not appreciated. And another thing we could learn is that the FOXP3 positive cells that we see that don't express RORC really did not come from this. So people have reported in mouse models or in vitro stimulation where you could stimulate a T cell in a certain way, it co-expresses FOXP3 and ROR gamma T, and then loses expression of one as it differentiates. And at least they may well be occurring, but in our tumor system or in our data, these cells that we see are clearly not arising from, or do not appear to be arising from these cells because they don't have really any significant amount of clonal expansion. So, in terms of biology, I think we've learned a lot through this paired analysis. And, given the negative association of IL-17 with cancer prognosis, people have proposed targeting IL-17 somehow in tumorigenesis under certain circumstances. But our data seems to indicate that a more logical point of targeting this pathway would be right here, before the horses have left the barn, so to speak, before you get this massive expansion, this massive IL-17 secretion. We want to figure out what is actually going on here, because this is probably a step you want to stop. And so this is basically analysis. I've done a lot of hand-waving based on one tumor sample, so I acknowledge that we really need to do a lot more analysis on this. And we know that tumors are very heterogeneous, but I think that this kind of shows what you can do by looking at multiparametric analysis and TCR sequencing at the same time. So that's that. I think I ended early. I'd like to thank Mark, obviously, and Mark and Chen, who are my mentors in the Davis lab. Jake is the computer guy who did the bioinformatics for this. And Leo is actually an oncologist from Germany who is doing a postdoc in our lab, who's helped with a lot of this analysis as well. And the genomics facility, G did all our sequencing for us. And I'll open it up to questions.